I'm oral historian Mike Chappell. Today, June 24th, 2012, I'm interviewing Dr. David Orth for the Endocrine Society at its annual meeting being held this year at the George R. Brown Convention Center in Houston, Texas.
Well, we were fortunate in developing an assay for ACTH because the structure-function relationships of ACTH were very well known. They'd been worked out primarily by Ciba-Geigy in Switzerland, who had synthesized all these analogs and fragments of ACTH and MSH, looking for uses for pharmacological agents. So using these fragments and analogs, we tested them with our antibodies, and we could tell very clearly exactly where our antibody bound to the ACTH molecule. Because of Ciba-Geigy's work, it was known precisely where the biological activity of the molecule resided. So ACTH is 39 amino acids long. It was known that the majority of the biological activity resided in the first 18 amino acids, and it was known that if you did very much in terms of changing those first 18 amino acids, biological activity disappeared. So it was absolutely apparent that the chances of developing a radioimmunoassay that would measure precisely the biological activity of ACTH was very, very small. And that depending on where the antibody bound to the molecule, the ACTH could last in plasma either a shorter time, about the same time, or a longer time than the bioactive ACTH did. And we did experiments to prove that. We took individuals, we suppressed their endogenous ACTH secretion with dexamethasone, then we injected them with synthetic human ACTH, and we followed them every few minutes by sampling blood and assaying biologic ACTH and ACTH by various radioimmunoassays. We were able to do this, by the way, in the biological activity because we injected a huge amount of ACTH, so we didn't have to have huge amounts of blood. And exactly what we predicted happened, that is that the immunoreactivity disappeared more rapidly than the biological activity, and the immunoreactivity disappeared at different rates depending on what particular antibody you were using. This was anathema to Gallo and Burson, who were quite understandably trying to establish the physiological relevance of these peptides, and they objected quite seriously to our findings, but eventually this was found with other peptides. As a matter of fact, Rosalind published data herself, and it became perfectly obvious that this was true of all peptide radioimmunoassays. Cushing syndrome is an interesting disease. It's one that occupied my interest for a whole 30 years. And with the ACTH assays and the MSH assays and later the beta endorphin assay, we were able to do a lot of interesting work with that. Cushing's, I think, is one of the most interesting endocrine diseases for a variety of reasons. Number one, it can be caused by pituitary tumors, corticotrope tumors. It can be caused by corticotrope hyperplasia. It can be caused by ectopic ACTH secreting tumors, tumors that are non-pituitary that make ACTH. It can be caused by ectopic CRH producing tumors, CRH being corticotropin releasing hormone. It can be caused by adrenal adenomas, benign tumors of the adrenal, by malignant tumors of the adrenal. It can be caused by macronodular and micronodular hyperplasia of the adrenal, both of which can be either sporadic or, particularly in the case of micronodular, hereditary. It can be caused by physicians giving patients steroids knowingly or unknowingly. Sometimes they're contained in drugs that are not always apparent. Cushing syndrome can be intermittent, it can be cyclical, and on top of all that, it's so rare that the average endocrinologist only sees 1 or 2 cases perhaps in his entire career. And so it remains a challenging kind of syndrome for endocrinologists to deal with. In our situation, we saw lots and lots of patients with Cushing syndrome, which was lucky for us. We were able to characterize in detail the nature of the disease. We saw patients who had unusual variations of Cushing syndrome, and were able to publish those and to explain their physiology and to elucidate how they fit into this overall picture. So I think without the ACTH assays, we wouldn't have been able to do that. And with the ACTH, MSH assays, and the ones we developed after that, they really formed the basis of all the things that I did in my lab for 30 years.